All right, everyone. Welcome back to Metabolism Part 2. My hope was that you had a chance to do the metabolism worksheet and bring it to class today. The one thing I want you to sort of like refer to it at the moment is on that worksheet, it asks you to draw a eukaryotic cell and diagram where the different aspects of cellular respiration are happening in a eukaryotic cell. And so let's have a look at it. So again, we're going to focus right now on aerobic respiration in a eukaryotic cell. And this should be, of all the things we're going to cover, hopefully the most familiar because you will have looked at it in cell biology and in A&P. And if you took nutrition, you probably had a quick peek at it there as well. So let's start with this, thinking about the eukaryotic cell. In which part of that cell is glycolysis, the process of glycolysis, that 11-step process that takes glucose and breaks it into 2 pieces of pyruvate? Where is that happening? Great. And if you don't know, you can put a question mark, but do put your answer in if you do know the answer. Fabulous. Okay, so most of you have said that it's happening in the cytoplasm, and that is true, okay, in the cytoplasm. Now, at the end of glycolysis, what you have is you've taken your 6-carbon glucose and you've broken it into 2 pieces, 3-carbon pyruvates. Pyruvate is then converted to acetate. Pyruvate is a 3-carbon molecule, acetate is a 2-carbon molecule, so that requires that you remove a carbon and throw it away as CO2. So that's the first place where CO2 waste is produced. But where does this process of pyruvate decarboxylation happen? So glycolysis happens in the cytoplasm. Where does this conversion of pyruvate to acetate happen? If you're not sure, you can put a question mark. If you think you know the answer, put in your answer. Don't be afraid to be wrong. And also just to give you a little bit of, hey, it's all good. This one step is one that took me ages to remember. I was always confused between where it was. So if you're not feeling confused, you're one step ahead of me when I was an undergrad. Good job. All right, so this happens actually in the matrix of the mitochondrion, which means that pyruvate has to be transported across the 2 mitochondrial membranes, a process that does require energy. Where is the Krebs cycle? So the acetate then goes into the Krebs cycle, right? It's a 2-carbon molecule. It goes into the Krebs cycle and is joined up to a 4-carbon molecule called oxaloacetate, becoming citrate, and then it goes through the cycle. Successively, 2 carbons are removed, electrons are removed, energy is removed, and we come back to the oxaloacetate ready to accept another acetate. Where does this happen? That's right, it happens in the mitochondrial matrix. The electrons that have been harvested in the process of the Krebs cycle, and a little bit in the pyruvate to acetate conversion and a little bit in glycolysis, but primarily in the Krebs cycle, that's where most of the electrons are removed from what was originally glucose. Those electrons are fed to the electron transport chain. Where is the chain, the actual components of this electron transport chain? Where are they located? Okay, good. Everyone has a little more confidence with this one, which is as it should be, the inner mitochondrial membrane, right? The mitochondria has 2 membranes. It's the inner membrane where this is located. So if you think about the big picture of aerobic respiration, which involves first glycolysis, then the Krebs cycle, then the electron transport chain, in which of these pathways is ATP generated? Great. Okay, so this is one of those wonderful things where there's no wrong answer, although there is a best answer. So it turns out all of them, right? Remember, glycolysis produces a net of 2 ATP. It actually makes 4, but uses 2, so it produces a net of 4. The Krebs cycle produces ATP as well, a little bit, a couple of ATPs. Majority of ATP is produced in the electron transport chain, right? Another thing that happens, because remember, I've been sort of mentioning this a couple times, but really the purpose of breaking down glucose to pyruvate to acetate and putting it through the Krebs cycle, the main purpose is to remove electrons. There's lots of electrons in glucose. We're removing them. Electrons are energy, so we're taking energy out, and we have to put them somewhere, right? We talked about last class how NAD plus is that little electron taxi cab, so they have to climb in and get taken to their next destination, which is the electron transport chain. So when is NADH produced? In which of these pathways? When do the electrons climb into the taxi cab producing NADH? Great. So it actually happens in all of the steps leading up to the electron transport chain. So in glycolysis, a little bit of NADH is produced. Some electrons are removed here. When pyruvate is converted to acetate, a little bit more electrons are removed and NADH is produced. Going through the Krebs cycle, the majority of electrons are removed and put onto NAD plus, making NADH. So the NADH is produced in all of these steps. And then it's oxidized back to NAD plus, meaning the electrons get out of the cab, right? This cab full of electrons has driven the electrons to their destination, and it's oxidized back to NAD plus. The electrons are delivered, the electrons get out. Where do they go? Right. The whole point was to take them to the electron transport chain. What I like to think of is taking them to the train station. I don't know why that's easy for me to remember, but I have a very vivid idea of these electrons like at some Broadway show getting into the cab and being taken to the train. Okay. Now, the point of taking electrons to the electron transport chain is so that they can be transported down the chain. And because the electrons are energy, as they move through the electron transport chain, their energy is removed, right? And it's used by the members of the electron transport chain, which are also proton pumps. And so that energy is used to actively transport protons across the membrane to form a gradient. So it's transporting them across the inner mitochondrial membrane. Where are the hydrogen ions, those protons, where do they accumulate to form a gradient in the eukaryotic cell? What's the part of the cell that becomes sort of filled up with these hydrogen ions that are being actively transported using the energy of these glucose electrons? That's right. That little space in between the inner and outer mitochondrial membrane is a safe place to stow these hydrogen ions, because of course, when you have a lot of hydrogen ions, what's true about the pH of that space? It becomes acidic, right? And the majority of enzymes in the eukaryotic cell, like a neutral pH, glycolysis enzymes do, this enzyme does, the Krebs cycle enzymes do. So you can't put those hydrogen ions somewhere that, you can't put them in the cytoplasm. You can't put them in the matrix. So you have to put them somewhere safe, and that happens to be that nice intermembrane space. Then what happens is that the gradient of hydrogen ions, that high concentration of hydrogen ions that has been created by active transport driven by energy from electrons that originally came from glucose, those hydrogen ions are allowed to come back passively across the membrane through ATP synthase. So ATP synthase has to be located in that same membrane with the electron transport chain, so like really close by. In a eukaryotic cell, it is possible to measure how much ATP is made per glucose. This number varies a little bit, and it's sometimes stated differently depending on the textbook you read. But in this class, what did I tell you when you did your prep? was the amount of ATPs per glucose in a eukaryotic cell? Right, 36, okay? So here's a little chart that kind of summarizes all this information. Like this is how I like to remember stuff that's kind of complicated like this, is to make myself little pictures. I'll draw you a picture in a second, and little charts. And so the eukaryotic cell, this is all this, where everything is happening and how much you get. In a prokaryotic cell, of course, glycolysis is still going to be happening in the cytoplasm. But if we turn our attention to that prokaryotic cell, some of these other things are not going to be the same. And that's because what is missing from the prokaryote that makes aerobic respiration so different? Yeah, there's no organelle, so there's no mitochondrion. And if you cast your mind back to chapter 4, that last thing we talked about right before the test, mitochondria are thought to have evolved from bacteria, prokaryotes. So prokaryotes are kind of like their own independently living mitochondrion. So they don't have a mitochondrion inside the mitochondrion, right? So glycolysis happens in their cytoplasm. Where does the pyruvate oxidation step and the Krebs cycle happen? Right. It also happens in the cytoplasm. And that just kind of makes sense when you think about it, because if you sort of think, oh, a mitochondrion is once upon a time was a bacterium, then the matrix part of the mitochondrion is actually once upon a time was the cytoplasm of a prokaryotic cell. Okay, electron transport chain in a eukaryotic cell is found in the inner mitochondrial membrane, where it is in a prokaryotic cell. Correct. Plasma membrane, because that's the only membrane you have available. Now, remember that one of the important features of aerobic respiration, the thing that makes it possible, is that energy from the electrons harvested from glucose is used to create a hydrogen ion gradient. You actively transport those hydrogen ions into a safe place where they can accumulate to high levels. That's the intermembrane space, the space in between the two membranes of the mitochondrion. But this space does not exist in a prokaryotic cell. So the membrane that you're moving protons across is the plasma membrane. On one side is the cytoplasm, on the other side is the extracellular space. Where are the protons being actively transported to? Where is that gradient accumulating? That's right. It's in the extracellular space. This also makes sense when you think about the origin of the mitochondrion, right? Because that outer membrane of the mitochondrion is actually thought to have been the plasma membrane of the cell that engulfed it, right? So the intermembrane space is actually once upon a time was the extracellular space of the prokaryotic cell. All right, ATP synthase is going to be in a eukaryotic, it's in the inner mitochondrial membrane. There isn't one of those. So ATP synthase is found where in a prokaryote? Right. Correct. Is found in the plasma membrane. And we got 36 ATPs per glucose in a eukaryotic cell. And how many do we get in a prokaryotic cell? Correct. We get 38. So one of the take-home messages you can say is that prokaryotic cells are better than you. But why? If you can explain, based on your prep for this section, why it is that a prokaryotic cell is actually better at making ATP than a eukaryotic cell, or gets more ATP per glucose, raise your hand. Aria, go ahead.
Is it because the pyruvate doesn't have to cross two membranes?
That's exactly right. So remember that because glycolysis happens in the cytoplasm, and the rest of the process happens in bits of the mitochondrion, the pyruvate that's created by glycolysis has to cross two membranes to get into the matrix. And that basically costs the ATP that was generated during glycolysis. Glycolysis pays for that transport. And then the rest of the ATP is made as a result of Krebs cycle and the electron transport chain. Okay, so let's do a little drawing really quick to illustrate this. So this is my drawing, which is why it is the way that it is. So you're say prokaryotic cell, here's glycolysis and the Krebs cycle happening in a prokaryotic cell's cytoplasm, generating a lot of NADH, right? That's gathering those electrons. The electrons are then dropped off at the electron transport chain, which is in the membrane. The energy from those electrons is used to pump protons outside the cell, and then those protons, by facilitated diffusion, come back in to create ATP. Eukaryotic cell different, of course, and here this diagram, the mitochondrion is shown unnaturally large for just to illustrate what's happening. We have our cytoplasm over here where glycolysis still happens. Then we've got our pyruvate that has to be transported into the matrix. We've got NADH production during glycolysis and during the Krebs cycle. And then the energy or the electrons harvested during all of these processes are dropped off at the electron transport chain, which is located in the inner mitochondrial membrane. And that oxidizes them back to NAD+, so they can go back and collect more electrons. Meanwhile, protons are pumped out into the intermembrane space. They come back through ATP synthase, and then energy is generated. And so you can kind of see there's a lot of similarities, but obviously it's a little more complex and a little bit less efficient in the eukaryotic situation, just because of this need to transport the pyruvate across two membranes. Okay, now that we've compared eukaryotic aerobic respiration to prokaryotic aerobic respiration, it's important. Now we're going to sort of focus on prokaryotes, right? So we sort of dispensed with our friends, the eukaryotes, because we've done them to death. Now we're going to focus on these prokaryotes, and we're going to compare aerobic and anaerobic respiration, because of course, as I mentioned last time, and hopefully you'll remember forever, these two forms of respiration are similar in that they both require glycolysis and the Krebs cycle and an electron transport chain. And the major difference between them is where the electrons go at the end. And anaerobic respiration is an option in some prokaryotic cells. So how are these similar? What did I just say? What is similar between them? What biochemical pathways do they share in common? Yes. Good. Yeah, that's right. All forms of respiration are categorized that way because they break down glucose, put it through the Krebs cycle, and then send the electrons that have been harvested to the electron transport chain. How do they differ? What's different between them? Great question, Leslie. No, they're not in reverse. They're parallel. It's just the differences at the end. And we're going to look at a diagram of that, and hopefully that'll make a little bit more sense. What you're thinking of, I think, Leslie, is aerobic respiration versus, say, photosynthesis. That's kind of a more accurate. Yes, Brittany. Yes, Heather. That's right, Jessica. Oh yeah, Heather, that's another good point. Yep. Okay, so there are two major differences between these pathways. In terms of sort of like the logistics of what's happening, the major difference is that aerobic respiration uses oxygen as the so-called terminal electron acceptor. So let's just unpack that unwieldy term for a second. Terminal meaning final, electron acceptor, where the electrons go. Remember, the whole point of respiration is you take glucose, which is packed full of high energy electrons. You steal those electrons out, you squeeze their energy out in order to make ATP, and then you can't just throw electrons in the trash. They have to go to another molecule. So we put them onto oxygen because oxygen is a very high electronegativity, meaning it pulls on electrons pretty hard and is willing to accept extremely low energy electrons. So in aerobic respiration, oxygen is the terminal electron acceptor, where after the electron transport chain has squeezed all the energy out of the electrons, or most of it, they drop them onto oxygen. But in anaerobic respiration, the electrons can't go to oxygen because that's just not how it works. It's anaerobic. So they go to something else. And an example of an alternative to oxygen that could work if you have the right enzymes is nitrate, but there's other ones too. So what we can say is that the terminal electron acceptor is different. And as Heather mentioned, the ATP yield is different. Okay, which of these processes produces more ATP? Correct. Aerobic respiration is always better. Anaerobic respiration is less than aerobic. Okay, so it's less than 38. How much less depends on specifically the details of what the alternative to oxygen is and which specific enzymes in the electron transport chain in that particular organism. But the ATP yield is always less than 38, whereas aerobic is always 38. Okay, so here is a little diagram to help us understand this. So here is respiration, okay? This is sort of a template for all respiration. We have glucose, 6-carbon molecule being broken down during glycolysis, 11 steps summarized by this arrow, into 2 3-carbon pyruvates, which are decarboxylated to produce 2 acetates. In the process, electrons are removed, and we create NADH. The acetate goes through the Krebs cycle, and it's got some carbons that are removed, ATP is made, NADH is made, and then all of these NADH molecules carrying electrons drop their electrons off at the electron transport chain, oxidizing them back to NAD+ so they can go back and collect more electrons. The electron transport chain pumps protons across the membrane. So you can notice that this is a prokaryotic cell because there's no mitochondria. It's all very straightforward. And then these protons come back through ATP synthase to generate ATP in the cytoplasm. Now, if this was aerobic respiration, who would be here to catch the tired electrons after they had moved through the electron transport chain and had all their energy used up? In aerobic respiration, who's here at the end to gather those electrons? Oxygen, that's right. Okay, and when you put electrons onto oxygen, it makes it temporarily negative. Remember, it creates that superoxide anion, which is not great. But luckily, there's a bunch of protons around and they can quickly quench that negative charge. And if that doesn't happen spontaneously, superoxide dismutase is there to make sure that it does happen. And so we produce water. That's aerobic respiration. Now, in anaerobic respiration, there's no enzyme that will do that. And so instead, the electron transport chain is modified. This is an example of one possible modification. You can put a different enzyme that is able to take those electrons and put them onto nitrate, producing nitrite instead. Okay, this is an enzyme called nitrate reductase. So this is an example of anaerobic respiration. There are other ones like carbonate producing methane or sulfate producing hydrogen sulfide gas. All of these alternatives are inorganic molecules that have a high electronegativity, so they're good at accepting electrons. However, and this is very important, none of them are as good as oxygen, which means they don't accept electrons that are as tired as the ones that oxygen accepts, which means you have to stop squeezing the energy out of the electrons earlier, right? You have to stop earlier. So there's not going to be as many times when the electrons are used to pump protons. The electrons are going to be less tired when they go to nitrate or carbonate or sulfate, but also fewer pumping activities have happened. So fewer protons are over here, which means less ATP is generated. Okay, so aerobic ATP yields 38 and anaerobic respiration yields what? Correct. Less than 38. We do know that it is not 0, okay? There is a minimum that it is going to be more than, okay? It's going to be more than the amount of ATP generated in glycolysis. because the total includes the glycolysis. And so we know that there's going to be a minimum, and it doesn't drop down to 0. So it's going to be more than 2 and less than 38. And you might be like, well, why isn't it more than 4? Because there's 2 that come through the Krebs cycle. Well, because some forms of anaerobic respiration have modifications to the Krebs cycle too. So, it's not for sure that you'll get all 4, but you do know that it's going to be more than fermentation produces and less than aerobic respiration produces. That part you can be sure of. So does that make sense? Yes, no, or kinda? Great. Okay, so that's sort of 50-50 on the yeses and the kindas and a few no's. So this is the material that you need to review. And also, if you have specific questions, you can stay after class and I'm happy to talk about your sort of like review parts of this that are still confusing, or even next class, we can do that. Or after any lab, if you want to talk to me, I'm available, or you can make an office hour appointment with me. A little heads up that I have only about 20 minutes after class today, because I do have another meeting that starts at 12:15. Okay, so let's move on for now. And let's do a comparison. So we've compared aerobic respiration in prokaryotes and eukaryotes. Then we've compared aerobic and anaerobic respiration in prokaryotes only. Now we're going to compare respiration and fermentation. So again, all the forms of respiration and all the forms of fermentation do share one biochemical pathway in common. And what is that? What do they share in common? That's right. They both use glycolysis, right? So glucose gets broken down to pyruvate every single time. What is the difference? What is the major substantive difference between all forms of respiration and all forms of fermentation? And don't say oxygen, because there's some respiration that can happen without oxygen. So what's the difference between respiration and fermentation on the whole? Or you might say, yes, that's exactly it. Okay. And if you're not sure, you can read the chat and see what other people are saying, because they're on the right track here. That's right. Yes. Okay, good. So major logistical difference is that respiration continues on to the Krebs cycle and the electron transport chain, whereas fermentation terminates at glycolysis. And the other difference, which is an outcome, is that the amount of energy generated by fermentation is always the same. It's only 2, and respiration is always more, whatever form it is, always more than 2. Okay, so fermentation stops making ATP after glycolysis. There are a few other reactions that happen. That's what determines which specific fermentation waste product you make. Is it acid? Is it gas? Is it alcohol? Is it an organic solvent? Is it acetone? But those reactions that happen after glycolysis, which are just 1 or 2 reactions that are unique to the type of fermentation, they don't produce any ATP. So the only ATP you make during fermentation is in glycolysis, and it is 2. Now, the other thing that you can sort of say about this, and this might be a little abstract, so I want you to buckle up your biochemistry seatbelt if it's not your jam. But again, this term terminal electron acceptor. In respiration, the terminal electron acceptor is either oxygen, if it's aerobic respiration, or an oxygen alternative like nitrate, carbonate, sulfate. All these things are highly electronegative inorganic molecules that can accept higher electrons. In fermentation, there is a terminal electron acceptor. For example, in lactic acid fermentation, the terminal electron acceptor is pyruvate itself. So we take electrons off of glucose and then we put them back on to pyruvate. It seems kind of crazy that that's how that works, but it is how it works. Other forms of fermentation, it might be putting them back onto acetate, but it's always putting them back onto an organic molecule. And organic molecules inherently have a lot more energy, and they only accept more energized electrons. So it's part of that whole thing about you're not getting all the energy out that you possibly could. Okay, so of the three options, aerobic, yeah, go ahead.
Question, sorry. So for respiration, is it inorganic or organic?
So respiration is an inorganic terminal electron acceptor. So oxygen is an inorganic molecule, sulfate, carbonate, nitrate are all inorganic molecules. And then in fermentation, the electron acceptor is pyruvate or a derivative of pyruvate. Pyruvate is an organic molecule. So it's always, fermentation
Okay.
always has an organic terminal electron acceptor. Does
Thank
that make
you.
sense?
Yes.
Great question. Okay. So now we're going to do a little picture here. Okay, so here's respiration, right? And we're sort of
I
extracting
think
it.
you skipped over your last bullet because I asked that question. Oh, never mind.
I know I was in the middle of saying something, but I think it was just to remind us of this little fact.
Oh,
here.
okay.
Yeah, and thank you for checking on that. I appreciate that. Yeah, so let's look at a picture. And again, just respiration is shown here, not in the context of a cell, but just sort of pulled out of it altogether, where glucose is broken down during glycolysis, then converted to acetate, then put through the Krebs cycle. Then the electrons go to the electron transport chain to create ATP. This regenerates the NAD+ molecule that's required to go harvest the electrons from all of these places. That's basically respiration. You know, the details of who is here to accept electrons, make it aerobic or anaerobic, or whether there's a mitochondrion or not a mitochondrion, make it prokaryotic or eukaryotic. But this is how respiration works. Fermentation goes to the pyruvate step, and then something else happens. But it doesn't proceed further to the Krebs cycle or the electron transport chain. So as an example of something that you will have heard of before, because this is what our bodies do, lactic acid fermentation goes like this. We break glucose in half to make pyruvate. We harvest some electrons out, but we're not going to use them in the electron transport chain. So we go, sorry, didn't mean it. I have to put them back somewhere. We drop them onto pyruvate itself, regenerating NAD+, so it can go back and harvest more electrons. And that creates another 3-carbon molecule called lactic acid, which has a reduced version of pyruvate. This is an alternative that you also probably heard of. Maybe you didn't know the details before, but yeast instead do ethanol fermentation. And what they do is they take pyruvate and they just go one extra step, right? They do that decarboxylation step, removing a carbon from pyruvate to make acetate. They drop the electrons onto this molecule instead, which is a derivative of pyruvate. It's still an organic molecule, to regenerate the NAD+, and that produces ethanol, a 2-carbon molecule that is the reduced form of acetate. And so this is known as ethanol fermentation. Now, there's not just these two options. There's all kinds of fermentations producing various types of acids, organic solvents, or alcohols. It just depends on which enzymes are present in that particular microbe to go the next step after glycolysis has happened. So here is a picture showing basically the difference between respiration on the left and fermentation on the right, and kind of indicating that what happens after pyruvate depends on the enzymes that an organism has. And just so that you have a sense of what those options might be, this is straight out of your video lectures. But different species or different genera, I should say, species, multiple species are in each of these genuses, but different genera that you've heard of now, right? You've heard of Clostridium, that's that rod-shaped anaerobe that includes Clostridium tetani, Clostridium botulinum, Clostridium difficile. Saccharomyces cerevisiae, you might not recognize this name, but this is the budding yeast that makes beer, wine, and bread. So you've certainly been exposed to the products of this organism. Streptococcus, right? Streptococcus pyogenes is the causative agent of strep throat. Lactobacillus, which helps make yogurt. Bacillus, which is a big genus that many of you are working on right now for your unknown ID. Escherichia, which includes E. coli. Salmonella, which can make you sick. etc. But look, they can all do forms of fermentation and they don't all produce the same end product, right? So these genera do lactic acid fermentation, just like us. But yeast does ethanol fermentation. And notice that it produces CO2, which is a gas, and that's why beer and wine initially is fizzy. This is my favorite one. I know I'm not supposed to have a favorite, but propionic bacteria produce propionic acid, and this is used in the production of Swiss cheese, which gives it a unique flavor. And so on. We could talk about these all night. Clostridium produces mostly organic solvents, which are nasty, and some gas. So just to give you the sense that fermentation is just not a one size fits all thing. However, there is something in common. All forms of fermentation produce the same amount of ATP. And what is it? Put that into the chat. Correct. 2 ATPs. Okay. I want to briefly, very briefly, discuss the catabolism of other macromolecules. So we've focused on carbohydrates. When you or bacteria eat a complex carbohydrate, it gets broken down into its component monosaccharides. Most of the time, especially because a lot of us in the macro world eat plants, and plants store sugar as starch. And so it's just a polymer of glucoses. So most of the sugar that we eat is actually glucose. But if you drink milk, it's got lactose, which is a disaccharide containing glucose and fructose. And if you eat fruit, you get fructose instead. If you eat cane sugar, then you get a disaccharide called sucrose, which is glucose plus fructose. Typically what happens with glucose or with sugar is it all gets converted to glucose so that it can all go into either fermentation or respiration. But we all know that us and prokaryotes can also extract energy from other macromolecules. So besides carbs, what are the other 2 major fuel sources for living things? That's right. Proteins and fats. And when we think about what's required, like in the environment, whether it's in the sandwich that a human being eats, or whether it's in the pond that a protozoan is swimming through or bacteria is in, primarily you're going to find fats and proteins in the form of proteins, which is a polymer of amino acids, and fats as triglycerides. So you initially do have to break down these macros into their component pieces with proteases and lipases. But after that, how does it all fit into the picture of respiration? So I want to show you that. So here's respiration. Here is glucose, which is where if you are eating carbs, they're going to sort of be converted to glucose. And then where in this process do the other macros come in? Let's start with triglycerides, okay? Triglycerides are 3 fatty acid tails on a glycerol backbone. And these fatty acid tails are either 16 or 18 carbons long, and the glycerol molecule is 3 carbons long. So the first thing you have to do is basically trim this all into different pieces. So like a butcher, you kind of trim off the 3 legs and then you've got the backbone. This 3 carbon molecule is actually very similar to pyruvate, so it gets fed in at this level right here. But these are 16 to 18 carbons long. They get chopped into 2 carbon pieces with that little cleaver. Chop, chop, chop. And then those 2 carbon pieces come in here and get fed straight into the Krebs cycle. Okay, so pyruvate, glycerol gets fed in about the level of pyruvate, and then these fatty acid tails are chopped into small pieces that end up feeding directly into the Krebs cycle. Proteins are polymers of amino acids. Each amino acid looks like this. It's got a central carbon, a carboxylic acid group, an amino group, and a mystery group. And we don't need the amino group. So the first thing that has to happen is we throw it away. And then that process of removing the amino group is called deamination. Microbes do it, you do it. Deamination produces ammonia. In our bodies, ammonia is very alkaline and extremely dangerous. And so it's immediately converted into urea and then excreted in the urine. But microbes don't necessarily do that. They often just throw it away and cause the environment to change pH. So here's the amino acid. The number of carbons in it is going to determine where it goes into this process, but the number of carbons depends on what this mystery group is. So if there's just a hydrogen here, then this 2-carbon piece can go basically into the Krebs cycle. But if it's, there's a carbon here, that's 3 carbons, it's probably going to go in here somewhere around the middle of glycolysis. And if there's more than that, 4, 5, or 6 carbons, there's lots of places in the Krebs cycle where those 4, 5, and 6 carbon molecules exist. So them different amino acids go in different places. And that's like part of the metabolism section of nutrition in like 30 seconds. So that's all you have to remember about that. And not in excruciating detail, but just to kind of understand that other macros are catabolized by trimming them down into conveniently sized pieces and putting them into some sort of convenient spot in the respiration process. The major reason I want to talk about that is to talk about exoenzymes. So what you may recall from digestion in human beings is that when we eat these things, like we go eat a sandwich and it's got carbohydrates in the bread and there's protein in the cheese and there's fat in the like mayo and the butter, you're eating macros that are long polymers of what you eventually can absorb. So you can't like just eat the sandwich and absorb the nutrients directly. You eat the sandwich and then your body, your digestive tract breaks down those macromolecules, the carbohydrates, into individual monosaccharides, the proteins into individual amino acids, and the triglycerides into fatty acids and glycerol. We do that in our GI tract. All along the GI tract, we have enzymes that do this. But a bacterium has no stomach. So, or it has no intestines, it has no stomach, it has no esophagus, it has no mouth, right? So it's going to use those digestive enzymes to break down those things, but with no stomach. And the other important thing to recognize is that, especially when it comes to bacteria, these single-celled bacteria, they don't have organelles like lysosomes. So the only way, and they don't have the ability to endocytose things. So when they're exchanging with their environment and bringing nutrients in, they can bring in one sugar or maybe a disaccharide. They can bring in one amino acid, but not a whole protein, because a whole protein would be the size of the transporter in the membrane trying to bring in the protein. So it would be like if you're trying to get a house in through the door of your house. So what they have to do is they have to break down the macronutrients in their environment outside. They have to secrete those enzymes out into the world and then wait for that stuff to come back to them or go chasing after it. So bacteria make exoenzymes. These are enzymes they're going to spit out into the world to digest the world, to make it more user-friendly for them. And that's because they don't have a GI tract like this, like we do, and they don't have lysosomes like a protozoa would. So what do we use? What does our body produce in order to break starch down into its component glucose monosaccharides? The enzyme that we produce it in our saliva initially. We produce a version of it in our pancreas that's then dumped into the small intestine. So it's pancreatic version. So there's the salivary one, and then there's the pancreatic one. Yes, it is the enzyme called amylase. So because bacteria don't have a mouth for saliva to be in, they don't have an intestine, they don't have a pancreas, they just make amylase and secrete it into their environment, and any starch in the environment will get broken down, and then those glucoses are now available for them to import one at a time. What do we use when we eat fat, which is usually in the form of a triglyceride? we need to break it down to fatty acids. What is the name of the enzyme category that breaks down fats for us? Correct. And so we have a little bit in our mouth, in the saliva, it's made under the tongue. It's called lingual lipase. It gets less as we age because we primarily need it to digest milk when we're breastfeeding. And then there's also some gastric lipase, maybe in the stomach, there's a little, and then pancreatic lipase. So we make lipase all the way along. And bacteria, of course, or prokaryotes, they secrete it into their environment to break the triglycerides up ahead of time so the fatty acids are more easily coming into the cell. We also eat protein, right? You eat a piece of cheese, or you eat some nuts, or you eat a steak. You need to break the protein down into its component amino acids. What is the class of enzymes that breaks proteins down? Correct, protease. And there's a variety of them. So some of them are, some of you mentioned specific ones that we know are in the human body, but proteases is the general category. Bacteria secrete them into their environment. Okay, so hopefully you're getting the picture here. All right, so the next thing we're going to talk about is those carbon fixation pathways, the autotrophs that that produce carbohydrates for all of the ecosystem. But it's pretty close to our break time, and I think this is a natural break point. So I'm actually going to have you come back at 11. So we get a little bit of a longer break today. Coming back at 11 to talk about photosynthesis, chemolytic autotrophy, and some lab-related stuff, which is pretty cool. So I will see you at the top of the hour at 11. Okay, so now that we've talked about these catabolic processes, how energy is generated by breaking down organic chemicals, let's talk about the processes that create the organic chemicals for the biological systems in the first place. So we're going to start with photosynthesis. And this is something that perhaps you have studied previously, but I asked you to look it over as your homework for today. In a eukaryotic cell, the process of photosynthesis includes the light reactions, okay, which are the photosystems that capture the photons of light, and the electron transport chains that use that light energy to pump protons across a gradient, across a membrane. So where are the light reactions happening in a eukaryotic cell? Correct. So they're happening on and around the thylakoid membrane of the chloroplast. The membrane, the thylakoid membrane is where the electron transport chain is located. So it's pumping protons across that membrane. There's going to be two sides to that membrane, the interior of the thylakoid and then the stroma, which is in the interior of the chloroplast. So which of those fluid-filled compartments is where we're actively transporting protons to create that gradient that we're going to use to make ATP? That's right. The thylakoid is a little pouch inside the chloroplast. And so we're putting the protons into the interior of the thylakoid. Now, the second step in photosynthesis is the light reactions generate two things, ATP and energized electrons. That is then put together along with CO2 to create sugar in the Calvin cycle, known as the dark reactions. Where are the chemical reactions involved in the Calvin cycle taking place? That's right, they take place in the stroma of the chloroplast. So again, we can compare that to a prokaryotic cell. because there are prokaryotic cells, such as photosynthetic bacteria, that do this process, but they don't have a chloroplast. So instead of thylakoid membrane being the location of the light reactions, where is it happening in the prokaryotic cell? Right, basically the plasma membrane. The dark reactions of the Calvin cycle that take place in the chloroplast stroma of the eukaryotic cell are happening where in a prokaryotic cell? Right, they're happening in the cytoplasm. And then we talked about where the protons are accumulating. They're being actively transported in a eukaryotic cell into the interior of the thylakoid, which is part of the chloroplast. That does not exist in a prokaryotic cell. So where are those protons going? We've got the electron transport chains that create the proton gradient in the plasma membrane of the prokaryotic cell. So we only got two options to put those protons outside the cell or into the cytoplasm. So where are they going? Correct. They're going into the extracellular space, just like would if this was respiration. Okay, so let's have a little eukaryotic prokaryotic comparison for photosynthesis. So here's the eukaryotic cell with an unnaturally large chloroplast. It's got its inner and outer membranes, and then there's going to be a thylakoid pouch diagrammed in here, right? And there's the thylakoid membrane and the interior of the thylakoid, and here is the stroma. The sun energizes electrons that have been stolen from water, which is where oxygen is created. Those energized electrons are fed through an electron transport chain, which uses the energy to make a proton gradient inside. The electron is then put onto an electron carrier for later use. Those protons are used to make ATP in the stroma, where the dark reactions happen, where carbon dioxide is put together along with the ATP that's been created and the energy of the electrons from NADPH. And that altogether creates a 3-carbon molecule. Now, if we compare to what's happening in the prokaryotic situation, all we have is basically plasma membrane, cytoplasm, extracellular space. So we've got our photosystem in here that is stealing electrons so that the sun can energize it. The electrons go through the electron transport chain located in the plasma membrane. Protons are pumped outside. The electron is then given to a new photosystem, re-energized, and then dropped off onto NADP, creating NADPH, because we're going to need this electron later to help make the organic molecule. Meanwhile, this proton gradient comes back through ATP synthase located in the membrane to create ATP in the cytoplasm. ATP plus the electron here, plus 3 CO2 molecules all together create a 3-carbon sugar, and 2 of those are put together into glucose. Okay. A couple of comparisons that are useful. If you're able to follow these, what it means is that you're starting to really understand these biochemical processes, not just at the nitty-gritty, I can draw a diagram and memorize all the steps level, but at the kind of much bigger picture level of what is the point of these processes in the first place. So again, the Calvin cycle is the dark reactions of photosynthesis. The Krebs cycle is this oxidation machine that takes electrons out of pyruvate. They're opposite of each other. Their purpose is opposite of each other, right? In the Calvin cycle, we are taking CO2 out of the air and we're adding electrons to it, and we're adding energy to it in order to make a high energy organic molecule. In the Krebs cycle, we're taking an existing organic molecule, in this case acetate, removing carbons, removing electrons, removing energy. So we can make a couple of comparisons. Let's compare these two processes to each other. In a eukaryotic cell, the Calvin cycle takes place where? In a eukaryotic cell, the Calvin cycle takes place in the stroma. Whereas in a eukaryotic cell, the Krebs cycle takes place in the matrix. That's right. So both of these are happening in what used to be the cytoplasmic space of the hypothetical bacterium from which those organelles are derived. In a prokaryotic cell, where are the Calvin cycle and Krebs cycle chemical reactions happening? That's right. They're happening in the cytoplasm. And so if we have a look at all these possible compartments, they kind of all have some things in common. The stroma and the matrix, the stroma of the chloroplast and the matrix of the mitochondrion are both pH of 8 fluids, and the cytoplasm is a neutral fluid. So it's all around the same kind of pH required for these processes to occur. Another comparison we can make between these cycles is whether or not they're overall oxidizing or reducing in terms of what we're caring about their products. So when we think about what's happening, let's start with the Krebs cycle, because that's one you're more familiar with. In the Krebs cycle, we are taking acetate and then we are removing electrons and putting them onto NAD+, making NADH. So would that make the Krebs cycle overall oxidizing or reducing? In terms of the pyruvate or the acetate that came into it, what's happening to the acetate? Is it being oxidized or is it being reduced? That's right. It's oxidizing in terms of the fuel that we're putting in the top, which is the acetate, which came from glucose. So it's part of the process of oxidizing glucose, taking electrons out of glucose. On the other end, the Calvin cycle, we're taking 3 CO2s so that we can have a 3-carbon backbone, adding some energy from ATP, and then NADPH is dropping electrons off to be used in bond formation. So is the Calvin cycle oxidizing or reducing? Right. We're basically reducing carbon dioxide so that we can make an organic molecule. So in the Calvin cycle, carbon dioxide is reduced. Electrons are added to it in order to create an organic molecule. And in the Krebs cycle, organic molecule acetate is oxidized to CO2. Electrons are removed from it. So the big picture of each pathway, the Krebs cycle is to break down an organic molecule to its lowest components and use the energy. The Calvin cycle its purpose is basically what we call carbon fixation, taking carbon, CO2, out of the air and creating organic molecules for use by biological systems. So we are in the carbon fixation realm now, right? And carbon fixation is really, really important for all life, right? There has to be in every ecosystem, there has to be a carbon fixing organism of some kind. Multiple ones are good. In our case, in the world that we live in, there's plants and there's algae and there's, bacteria, all of which do carbon fixation. But that's not the whole story. There's other parts of the world that maybe we aren't as familiar with because we don't tend to live there. Instead of photosynthesis, where sunlight energy drives the production of glucose, what's the other process? Something with rocks, yes. Chemo something, yes, you're both on the right track. Keep going, push, push. Chemolithoautotrophy, yes. Chemo is a word that means chemical. Litho is a word that means rock. Autotrophy is basically a way of saying carbon fixation. Autotrophy means we're making our own organic chemicals. We're not stealing them from other organisms. So how does this work? Here's a little cartoon. It kind of illustrates one possible form. There has to be some kind of chemical in the environment that it's easy to steal electrons from. And so a common one is iron or another metal ion that has an easily stolen electron. We stick that electron through an electron transport chain. Its energy is used to pump protons across the membrane, creating ATP. And ultimately that energy plus the electrons plus CO2 in the air is used to make a 3-carbon organic molecule, and 2 of those put together makes glucose. So this part varies between the different organisms that do cumulative autotrophy, depending on what their electron source is, but it's always some sort of inorganic environmental molecule like a metal ion. But this part here is extremely similar to photosynthesis, right? This major difference is right here. And so the importance of these carbon fixers, why do we have ones that don't rely on sunlight? What's the purpose of these organisms that can do this? Most of them, all of which are bacteria or prokaryotic archaea. Yes, and it is right. So that life can survive, and Sunday is right. So life can survive in areas where there is no sunlight, such as ocean floor, underground, like deep in the soil, inside a cave, et cetera, et cetera. Okay, so these are the carbon fixers of the dark. And here's an example of an environment where they might be important at the bottom of the ocean floor. Okay, so I want to turn our attention to sort of lab relevant stuff that you still need to know, but kind of tie it into some of the things that you're learning in the lab. Different organisms, different microbes, different bacteria have different metabolic strategies and abilities, which comes down to which enzymes they have. That means they can thrive in different environments. And it also means that if we have an unknown that we're trying to identify, or we're trying to characterize, we can run various tests to try to figure out what their abilities are. And if we're trying to identify them, who they might be. So remember that we've already talked about some of these metabolic tests. Here's the thioglycolate test, right? What's the purpose of this test? What does it tell you? Yeah, it tests for oxygen preference or oxygen requirement. Does it need oxygen? Does it not need oxygen? Can it use oxygen? Can it not use oxygen? Is it killed by oxygen, right? So here we've got an obligate aerobe and an obligate anaerobe. Who's this guy in B? Yeah, he's a facultative anaerobe. And here over here, we've got an aerotolerant guy. And just to be clear, like in real life, these two are not always so clearly distinguished. So there can be an organism that grows a little better at the top and okay at the bottom, and it can be a little hard to see that difference. So sometimes they can be confused for each other. But what you can certainly say is that these two types of organisms can thrive in both oxygenated and non-oxygenated environments. However, when we do figure out which one's which, and we say, okay, this one's definitely aerotolerant, this one's definitely facultative anaerobe, then that has implications for things that we can then infer about their enzymes. Okay, so starting with, here we've got our 4 categories. Well, there's a 5th category, but we're kind of ignoring the microaerophiles for now. Which of these can perform aerobic respiration? Put their letters into the chat. Which of them can perform aerobic respiration? Oops, I just gave the answer away. Let me take it away. It's now in your subconscious mind. Lock your answers in now. That was my cat. Dangerous having animals in the house. Correct. So obligate aerobe, of course, because it requires oxygen, and the facultative anaerobe is capable of it. And we know that it's capable of it because we can see clearly a difference in growth when the oxygen is present, when it's not present. Why does it grow better when it's in an oxygenated environment and doing aerobic respiration? Why is it growing faster here? Okay, oxygen's more efficient, it prefers oxygen, there we go. aerobic respiration produces more ATP, right? Produces more ATP than anaerobic metabolism, whether it's fermentation or anaerobic respiration. Okay, so what about which of these is capable of doing anaerobic metabolism? Great. A, B, and D. The only guy who can't do any form of anaerobic metabolism is this guy. Now, I'm using this word metabolism rather than respiration because metabolism encompasses all the possible ways that you can survive with no oxygen. If I was to ask you which of them can do fermentation, the answer actually would be the only thing you can be sure of is that this guy does not. Why is it that we can't say for sure that these guys that are growing without oxygen are doing fermentation? Is there any other reason they could be surviving in these anaerobic environments if they were not doing fermentation? Is there any other form of metabolism besides fermentation that allows an organism to survive without oxygen? That's right, Laura. Okay. That's right, Aislinn. They could be doing anaerobic respiration. So when you see something growing down here, you don't know exactly what it's doing. All you know is it's doing either some kind of fermentation or some kind of anaerobic respiration. And there's multiple forms of both of those. So you just know it's not doing aerobic respiration. You don't know what it's specifically doing. Okay. Now, this answer does have a definitive answer. Which of these has the enzyme superoxide dismutase? This is something you just had to memorize from the growth section. So you're going to have to know it for the test. Right. So all of you were certain about B and C. Some of you were iffy about A, but it is in all organisms that live in oxygen, whether they use it or don't use it, but if they're alive in oxygen, they have superoxide dismutase. Now, when it comes to catalase, which is another defense against reactive oxygen species, we actually can say for sure, of course, this guy doesn't have catalase. We also know that aerotolerants don't have catalase. That's just something you had to memorize. But we think maybe these guys have catalase. We don't know. And the reason we don't know is because there's an alternative catalase that performs the same function called peroxidase. So we don't know whether they have peroxidase instead. Okay. What about cytochrome C oxidase? Well, I haven't told you this yet. If you were to guess, you would probably say, well, cytochrome C oxidase is involved in the aerobic respiration electron transport chain. So probably that's going to be B and C, and definitely not D and A. You'd be mostly right. Because as it turns out, cytochrome C oxidase, which is has the job to take electrons from cytochrome C and put them on oxygen. There are a couple of enzymes with a similar function that could substitute in different microbes. So we don't know if cytochrome C oxidase is the one doing it. So if you get a negative oxidase, it doesn't mean they're not doing aerobic respiration. That's why the oxidase test is not a good proxy for oxygen requirement. Okay, nitrate reductase, part of anaerobic respiration. We know for sure who does not have it. Who can we be sure does not have it? That's right. We can be sure that C does not have it. We can guess that maybe A, B, and D could have it, but we can't be sure. So this is one of many forms of anaerobic respiration. If they didn't have nitrate reductase, what could explain that they're growing here? Put into the chat some alternatives to nitrate reduction as their method of surviving with no oxygen. Yeah, fermentation, or some other form of anaerobic respiration that it doesn't use nitrate, but instead uses sulfate or carbonate, etc. Alrighty, so let's ask a few questions about metabolism that are applied and see if we can understand them. Again, this is prepping you for the test. We've got a mesophile and we've got another mesophile, so they both like the same temperature. We grow one tube of each species in an oxygenated environment. One of them is a facultative anaerobe and the other is aerotolerant. So we can assume they're not fastidious. We can assume that they're capable of growing in this medium. Which tube most likely represents the facultative anaerobe? Yes, there are 3 tubes. One of them is a distraction. So which tube represents the facultative anaerobe? They're grown in oxygen. Great. So everybody has chosen B. Please raise your hand if you can defend your answer, which is the correct answer. Tell me why you chose B. Sunday, go ahead.
I chose B because you can see growth throughout the whole thing, but on the top you can see a little bit more growth.
Oh, thank you for that answer. Let me clarify something that is not clear. These are not thioglycolate deeps. These are just broths. So this is just a trick of the light. What you can see is that there's more growth here than there is here, and this has no growth at all.
Oh, okay. So then it would just be because there is more growth in B, like it's not as transparent, I guess you could say.
And why, and you don't have to have the answer to this, but if you do, let me know. Why does it have more growth than the aerotolerant one?
Because it can grow in the oxygen, or I don't know.
They're both growing in oxygen, but what's different about the facultative anaerobe when it's exposed to oxygen that the aerotolerant is not doing?
I'm not sure.
That's a great answer. Aislinn, did you want to help pick up the thread here and move forward?
I can certainly try. I think it's because the aerotolerant, it can't grow in that high a level of oxygen.
That's a great guess. That's actually not
Okay.
accurate, but you're actually sort of on the right track there. It is in fact growing in oxygen, but not as well. And let's see if Laura has an answer to why. Let's keep this going, see if we can get there. Laura, go ahead.
I think I remember you saying that the aerotolerant doesn't use oxygen. It just doesn't mind it being there.
That's right. And so why would that make a difference? How come what's the facultative anaerobe doing when oxygen is around? What metabolic strategy is it using?
Aerobic
respiration, right? And this guy is doing something else. So how come that leads to more growth? What's different about aerobic respiration versus any form of anaerobic metabolism?
Aerobic is the best.
In terms of creating more
ATP.
Correct. And the amount of ATP is what drives growth, right? The speed of replication. Does that make sense? Yes, no, or kinda, guys?
Dr. Hill, can you say that one more time? The amount of ATP drives the growth?
Yes.
Is that?
Yeah. So one of these tubes is inoculated with a facultative anaerobe, and the other is inoculated with an aerotolerant organism. They're both growing in oxygen, but the aerotolerant one is not using the oxygen. It's doing some kind of, it's either fermentation or anaerobic respiration. Whereas when oxygen is present, facultative anaerobes will switch to aerobic respiration, and aerobic respiration produces 38 ATPs per glucose, whereas this guy is making less per glucose. And so because there's more ATP available, they're able to divide faster, and the thicker, cloudier culture is the result of there being more cells that have been produced in the same amount of time. Does that make sense?
Yes, thank you.
Sweet. All right. Great questions and answers, guys. All right. So this is an example of the kind of question you might face on the test towards the end, where you're sort of asked to apply your knowledge, right? To figure something out based on the type of information. So, you know, if this is still kind of murky, just know this is something to work on. Okay. Now let's do another, we're going to use the same pictures, but we're going to propose a different scenario. So let's say we have a facultative anaerobe, and that's the only organism. We put it into two tubes. One will be grown with air and one will be grown with no air. Which of these tubes represents the tube that was grown with no air? Put your answer into the chat. Great. A lot of you are picking A, which is the right answer. If you are able to explain why you think so, you don't have to be right, but tell me what your reasoning was. Raise your hand. Why is A the right answer? Aislinn, go ahead.
Because without air, it's not going to produce as much ATP as it would with.
Right, because what's it not doing?
It's not using oxygen.
In the process of
Anaerobic respiration or aerobic respiration.
right, exactly. Because here's our two tubes, and again, this one was provided as a distraction. We've got the tube that was grown in air, and we've got the tube grown without air. We know that this is the one grown without air because there's fewer cells, and we know that because a facultative anaerobe, when provided with oxygen, is going to make more ATP per glucose, so it's going to be able to grow faster. Does that make sense? Yes, no, or kinda? Great. Okay, so I just want to sort of like connect this to the tests that you're learning about in lab and that you're going to be using for your unknown ID. Because in lab, we're going to just touch on like maybe 6 tests. But in the unknown ID process, you're going to probably do a dozen. And some of them you won't have like specifically learned how to do in the lab. But on the process of identifying your unknown, you're going to run a variety of metabolic and growth tests. Here are the ones we actually have. We have tests for growth at various temperatures. So you would just then get if you asked for what is the result for growth at 15 degrees, you'd get a picture of a tube and you'd see whether it was cloudy or clear, right? Did it grow or not grow? We have tests for growth at various pHs. Again, it's a broth that was set to a particular pH. We put some cells in it. We just see, did it grow or not grow? So you could say, what's the growth result for my at pH of 4? We've tested for growth at various salinity. Okay, so percentages of salt. So what's the growth of my organism at 15% salt? Is the tube clear or cloudy? If you want to know oxygen preference, you can do a thioglycolate test, as we hopefully I've beat you to death with that one. If we want to understand if it has specific enzymes involved in metabolism, like catalase or oxidase, we can do these tests. We're learning how to do that this week in the lab, but I tried to emphasize that these tests are not sufficient to identify oxygen preference for various reasons. Okay, hopefully some of those reasons now have become clear, because you know, a catalase negative doesn't mean can't do aerobic respiration, just might mean they do have peroxidase instead. And a negative oxidase test doesn't mean they can't do aerobic respiration, because there are some alternatives to cytochrome C oxidase. We also have a nitrate test. This is a very commonly used test to distinguish groups from each other and species from each other. And we're learning how to do that in lab this week. But of course, this is not definitive to say definitely, like if it's negative, doesn't mean it's unable to do any form of anaerobic metabolism. Sulfate reduction test, we have that too. So these are tests that test for various forms of anaerobic respiration. And this sulfate reduction test is not called sulfate. It's called Kliger's iron agar because it's named after some guy, okay? So you'll find this one in your microtest reference. We have tests for a variety of exoenzymes. Does the organism have the ability to secrete amylase, which is the starch test? Does it have the ability to break down casein, which is a milk plate? Does it have the ability to make lipase, which is a lipid plate? We have a huge schwack of tests that allow you to test for individual carbohydrates. Can you ferment them? Can you ferment sucrose? Can you ferment xylose? Can you ferment trehalose? We're doing some of them this week, but they're all kind of similar in their appearance. So just to kind of remind you what it looks like when you do catalase testing, and this is what you saw or will see in lab this week. When you do catalase testing, it either bubbles, which is positive, or it doesn't bubble, which is negative. Okay, so now I've told you that catalase is not a sufficient test to distinguish oxygen preference, but if you already knew something about the organisms, you could actually make conclusions. So let's suppose you have a facultative anaerobe and an obligate anaerobe. Okay, let's suppose you have a facultative anaerobe and an obligate anaerobe, and you do the catalase test, and one of them is in one of these samples and one is in the other. Which one is the one that bubbled? Great. So many of you are saying that A is the one that bubbled and that B must be. So this must be the, sorry, A is the one that bubbled. A is the facultative anaerobe. The one that bubbled was the facultative anaerobe. And B is the obligate anaerobe. How do we know that? How do we know that the obligate anaerobe, for instance, definitely doesn't have bubbles? Raise your hand. What do you know about obligate anaerobes, generally speaking, in terms of their ability to make all these reactive oxygen species defense enzymes? Aria, go ahead.
Obligate anaerobes don't do aerobic respiration, so they don't need defenses against oxygen.
Right. So, and more to the point, because an aerotolerant doesn't do it either, but still has a defense, but doesn't have catalase as a defense. But this organism does not normally live in an oxygenated environment. Therefore, it has none of the oxygen defense enzymes. It has no catalase, it has no peroxidase, it has no superoxide dismutase. So if you knew that one of the organisms gave bubbles and the other didn't, and you knew that one of them was a facultative anaerobe and the other was an obligate anaerobe, you would be able to tell which of these samples was which. And just for the record, what's being released here is oxygen, and it's not because of oxygen in the environment directly, it's because the catalase test is performed by putting cells on in a puddle of hydrogen peroxide, and hydrogen peroxide, when it's broken down by catalase, produces oxygen gas as a waste. Okay, now here's the oxidase test. Again, you will do this in lab, or you have already done it in lab. So a positive result on this test is like a super dark purple. A negative looks sort of barely purple or not purple. So this is kind of more negative. And some of you saw this yesterday in lab and were initially fooled by this, thinking that this was positive. But this is what a positive looks like. Something like this or lighter is a negative. So you have an unknown. If it gives you a positive result, what can you conclude? A negative is harder to interpret, but if an unknown gives you a positive result, what do you know about it? What's one thing you can say for sure? It uses oxygen as a terminal electron acceptor in the process of aerobic respiration. Exactly. Okay, so a negative doesn't actually tell you that it doesn't do aerobic respiration. That's because cytochrome C oxidase, which is the specific enzyme being tested for here, can be replaced by other enzymes in some organisms. But if we know that it does have it, well, that says for sure it's going to be able to do aerobic respiration. Okay. The other test you're doing in lab this week is the nitrate test. And so here's 4 different tubes with 4 different species, some of which you're actually going to do in class. There's E. coli, Pseudomonas aeruginosa, Staphylococcus epidermidis, and Streptococcus pneumoniae. And so this is going to be easier to understand once you actually take in the lab. So Tuesday people are sort of going to have a jump on this one. Sorry, Thursday people. But the nitrate test is this two-step test. And in the first step, you can turn red or stay clear. And in the second step, you can turn red or stay clear. The first step is two reagents that are sometimes called nitrate 1 and 2, although in lab you'll also learn that their other names are sulfonilic acid and alpha-nephthalamine, which thankfully you don't have to memorize for a test. The second step involves addition of zinc and hydrochloric acid. So we have tubes here that are red in step 1, clear in step 2, or red in step 2. Those of you who were here on Tuesday in lab know that this one on the end here that's red after step 2 is positive or negative. Yes, this one on the end is red after step 2. Positive or negative? That's right. The reason for that is the step 2 reagents turn red in the presence of nitrate. So what this test does is you offer nitrate, a broth that contains the chemical nitrate, you offer it to the organism, you wait a couple days to see what happened to the nitrate, and then you test for it. So in this case, we've put in chemicals that react with nitrate to turn red. And so this red color is saying the nitrate is still present. And so it was not reduced, it still remains. Over here, you have an organism. Well, let's do these ones in the middle. These turned red in step 1, right? Red in step 1 means what, Tuesday people? Red in step 1 is positive or negative? Right, red in step 1 is positive because the chemicals added in step 1 test for the reduced product of nitrate, which is nitrite. So they turn red in the presence of nitrite, which indicates that there is in fact nitrate, and nitrate has been reduced to nitrate. Now over here is another positive result. It is clear, or it can be gray or green, as some of you mentioned, or cloudy after the second step. So what that means is when you tested it for nitrate, when you tested it for nitrite, there wasn't any. But then when you tested for nitrate, there also wasn't any, suggesting nitrate was converted to something else besides nitrite. So that will make more sense if you didn't follow that, it'll make more sense in lab. But I'm just pointing out that the nitrate test is relevant because nitrate reduction is part of one form of anaerobic respiration. All right, exoenzymes are also something you can do. We won't do it in lab, but it's a test you can ask for. This is a milk plate, tests for the enzyme caseinase. You can see that there's 2 organisms have grown on this plate. The plate is milky because it contains milk. This one has no clearing around the streak. This one has a clearing around the streak indicating the milk has been broken down. Which of these organisms, left or right, contains the enzyme caseinase? Casein is the protein in milk that makes it milky. That's right, on the right. Now, this is actually a differential plate. It's differential for caseinase production. And you can see it's not a growth assay because both these organisms grew. It is a differential plate. Some visible difference, in this case, it's not color, but instead is clearing around the streak, indicates something about the organism's metabolic abilities. And again, why do bacteria need exoenzymes? Why are they secreting this and causing a chemical reaction outside of themselves? Well, it's because they don't have a GI tract or lysosomes or endocytosis as an option. This plate is mislabeled. I stole this on the internet and I only recently realized that it's actually backwards. But again, the lipase plate is one you can do. What it is it's a blue dye that's a lipid-based dye. So if the organism is capable of secreting lipase, it's going to clear the dye around the streak, another differential plate. And which of the organisms, top or bottom, is the one that has lipase? Right. Contrary to the labeling here, which is wrong, it is this one. This is the positive result showing that the organism has lipase and this one does not. Okay, the starch test is basically a plate that contains starch, and the starch plate is clear. The way to tell if starch has been broken down by the organism, because in this case you can see there's something that grew here and there's something that grew here. The reason this plate is dark right now is because in order to find out what happened, iodine, which is dark colored, was added to the plate. Iodine turns blackish brown when it's bound to starch. So which of the organisms on the left or the right was able to secrete amylase into its environment and break down the starch? Exactly. Just like it says in the text here, this guy on the left was secreted the enzyme. Because it cleared the starch, when the iodine came along, it had nothing to bind to. And if you ask for this test, you'll usually get two pictures. And the first picture will be the plate before iodine was added. And the next picture will be when it was added. Okay, so just a reminder that you really want to get your step 2 done as soon as possible. The theoretical deadline is this weekend, although it is flexible. And if you have questions, you could stick around or ask me in lab. That's an incorrect date, sorry about that. But the reason you want to get this done sooner rather than later is that step 3, which happens next, is going to take way more time. And we'll talk about that, how to do step 3 next week. Looking at our time, we have 3 minutes. So I'm just going to take these 3 minutes to really quickly show you where the step 3 stuff is in. And to also remind you what is going on this week in terms of what you need to be doing. Oopsies. There. So I have extended the growth quiz to Sunday because we didn't finish growth until Monday. But you also have the metabolism quiz this Sunday. You also have to do prep for lab 6. You also have to prep for the genetics section, which is coming up. And the genetics section is dense and fascinating, but heavy, and I encourage you to get a jump on that. So definitely do take the time to do all that stuff. Lab 4 and lab 5 are both due this Sunday. So you'll have by the end of lab this week, you'll have everything you need for lab 5. And if you were here last week for lab on Tuesday, you have everything you need for lab 4. If you were not, basically for the Thursday people, I'll reiterate this on Thursday in lab, but really the only part of lab 4 that I'm grading on anyway is the online stuff. Right? There was an online activity for you to do with growth tubes and thioglycolate and stuff like that. So if you have questions about that, whether you're in Tuesday or Thursday, you can stick around. But everybody's got everything they need to complete both these labs once tomorrow has happened for the Thursday people. Okay, so quick, just want to show you and remind you, if you are done with step 2, and some of you are done and have been graded, it is really important that when you go to start part 3, you read the instructions and watch the video. So read the instructions and watch the video. And then you may have questions, and that's fine. But if you don't read the instructions and watch the video, then your questions won't be as useful to you as they possibly could be, okay? You will have missed some basic information or a starting point from which to ask really helpful questions. All right. So I think I'm going to stop it there, and I'm going to stop the recording, and I'll take any questions that you have.